thank you, Jackie, and the Board of Directors of the Epilepsy Foundation and the organizers for the opportunity to speak to you all today. By way of disclosure, I am a full-time employee of CODA Biotherapeutics. I'd like to also thank the folks who did all the work who are here today. So I'd like to introduce them all to you and ask them to stand. Dr. Steve Dodson, Nadia Khan, and Ali Nassab, and Peter Klein from Ivan Soltes's lab, along with Quinn Ann Nguyen is our collaborators for the work I'm going to show you. So I'm going to talk to you today about chemogenetics. And as you might infer from the title, chemogenetics is a combination of chemo or chemistry, small molecule chemistry, and genetics or gene therapy. And CODA's approach to this is shown schematically on this slide. So what we've engineered is an inhibitory ion channel. This is a chimera of the acetylcholine receptor, ligand binding domain, and the glycine receptor ion channel pore. And so when it's open, it's actually going to flux chloride into the cell, increase the input resistance, essentially a chloride shunt that makes it less likely that the neuron is going to fire action potential. So it quiets the neuron. And importantly, it's activated by an oral small molecule. This would be acetylcholine receptor agonists that have a long clinical history to them. And it's important to recognize that neither of these components is active when they're dosed alone or when they're actually expressed alone. They're only going to reduce neuronal firing when they're together. So we've demonstrated in preclinical studies tunable control, not only at the single neuron level, but also at the tissue circuit level. And so on the left side, you can see hippocampal neurons that have been growing in vitro for 17 days. They're actually firing action potentials that you can see in that bottom trace, very frequent baseline action potential firing. And as we dose the drug, increasing concentrations of the lead drug bind to the receptor, open the channel, flux chloride into the cell, and reduce the likelihood of action potential firing in a dose-dependent manner. And this can wash out this effect. Inhibition of the action potential firing actually washes out. And importantly, we've also shown that at the tissue level. So on the right-hand side, you can see a hippocampal slice that's been prepared from animals that are either expressing the CODA receptor or a scrambled control. And time is on the x-axis with the number of action potentials on the y-axis. And what you can actually see is that at time 1, when a single dose of the CODA lead drug is given, we see a rapid inhibition of the number of action potentials in the animals that are expressing the CODA receptor, whereas there's no effect in animals expressing scrambled controls. Tunable control at cell and tissue levels. And so what about the seizure models? So this is the electrically induced seizure threshold model. This is something that you saw previously in Julia's really nice talk. This is a model where a probe is put into the brain and currents passed along the probe. And at a certain point of current amplitude, it actually triggers a seizure to occur. And so the higher that threshold is, the less likely the spontaneous seizures are to occur. And so you can see the design in in black, the animals expressing the CODA lead receptor versus orange, the animals expressing the scrambled control, treated with either placebo or the CODA lead drug. And it's only in the case of the animals expressing the CODA receptor and treated with the drug that we actually see an increase in the threshold for spontaneous seizures. The lead drug has no effect in control mice. And these effects are statistically significant by ANOVA and students' t-test, A and B respectively. We also looked at another model, the canic acid seizure model. Many of you are familiar with this. So here, canic acid is injected, an excitotoxin is injected, seizures are established in the mice. And then at some time later, the mice are injected with either the adenovirus expressing the CODA receptor or the scrambled control. And then after that's allowed to express maximally in the neurons, we treat the animals with the CODA lead drug or placebo. And it's really only in the case of the animals expressing the receptor and treated with the CODA drug that we see the inhibition of seizures. And the electrogenic traces are shown here for all 4 groups. And you can see it's only in the case of the 2, the receptor expression and the drug treatment, that we see a significant reduction in seizure frequency. These traces are taken from the graph shown here, where you can see each of the groups in turn, the control, scrambled control expressing animals in orange, and then the CODA lead receptor expressing animals in gray and black respectively, treated with placebo or CODA lead drug. And so time is again on the x-axis. We collect a baseline, and then at time 0, a single dose of drug is given. On the y-axis, we're looking at the number of seizures. And once again, it's only in the case where we have the receptor expressed in these animals and the drug on board that we actually see a significant reduction in seizure frequency. The drug has a fairly short half-life in rodents. It's only about an hour. It's much longer in humans. It's 23 hours, which is consistent with once daily dosing of the drug. But this is a significant reduction in seizure burden that we think actually bodes very well for the impact on total seizure burden in these animals and in the patients. So we took a look at the relationship between receptor expression and the number of seizures that the animals were experiencing when they were treated with drug. And you can see just by looking at it, we've got 2 animals shown here in A and B. You're looking at GFP expression, which is co-expressed with the receptor in the dentate gyrus. And you can see with your own eyes that these 2 animals actually have a different level of gene expression. And so those same 2 animals are marked on the graph to the right, where the GFP intensity is shown on the x-axis and the number of seizures on the y-axis. And what we see when we look at this is that the 2 are highly correlated with the levels of receptor. The more receptor that's expressed, the lower the seizure frequency that we see, with some animals actually achieving seizure freedom. So this quantitative relationship between expression and efficacy will allow us to really guide dosing and further development and identify where we really need to be in terms of tissue transduction. And we think represents an exciting possibility for the patients down the road in development. So thank you very much.